Geachte collega's, beste aanwezigen, dames en heren. Dear colleagues and guests, welcome to all of you. Please be seated. Van harte welkom op deze openbare verdediging van het proefschrift, getiteld Identification and Investigation of Probiotics for Oral Biofilm Management door de heer Wannes Van Holm. De kandidaat opteert ervoor om de verdediging in het Engels te voeren, vandaar dat de rest van deze zitting in het Engels zal verlopen. This public defense is the last requirement to be completed successfully by the candidate in order to obtain the doctoral degree. First, let me introduce you the candidate. In 2018, Mr. Van Holm graduated cum laude as Master in Biology at KU Leuven. That same year, he started a joint PhD project at KU Leuven in the research group Periodontology and Oral Microbiology of the Department of Oral Health Sciences, and at UGent in the Department of Biotechnology. Wannes' research work results today in the defense of his dissertation, and when preparing a PhD thesis, guidance by a promoter is essential. He has two promoters. The promoters of the research work presented here today are for KU Leuven Professor Wim Teugels from the research group Periodontology and Oral Microbiology of the Department of Oral Health Sciences. And for UGent, Professor Nico Boon of the Center for Microbial Ecology and Technology of the Department of Biotechnology. The promoters are guided the candidate of the last few years and were assisted by co-promoter Professor Christel Bernards of the research group Chemical and Biochemical Reactor Engineering and Safety of the Department of Chemical Engineering at KU Leuven. With pleasure, I also would like you to introduce the members of the examining committee. They are, of course, a very essential and important part, mainly in this final stage of the PhD training. Dr. Alex Mira from Genomics and Health Department at the Fisabio Institute in Valencia, Spain. Welcome to you, Dr. Mira. Professor Bastiaan Krom from the Academic Center for Dentistry in Amsterdam, better known as ACTA, in the Netherlands. Thank you for joining us here, Professor Krom. Professor Tom van der Wielen from the Center of Microbial Ecology and Technology of UGent. Professor van der Wielen, also thank you for joining us here today. And then from KU Leuven, I would like to welcome Professor Lieve van Mellaert from the Laboratory of Molecular Bacteriology of the Department of Microbiology, Immunology and Transplantation, and Professor Jan Michiels from the Microbial and Plant Genetics Units of the Department of Microbial and Molecular Systems. Also, thank you both for joining us here today. Let me finally explain you briefly the course of the public defense. The candidate will be invited to present his research work, and that within a time frame of about 30 minutes. After the presentation of the research work, the members of the jury will be invited to question the candidate. And also the audience will receive the opportunity to do so. At the end, the examining committee will retreat for the deliberation, and I especially would like to draw the attention of the jury members and also the audience to the fact that the regulations of our university prescribe explicitly that jury members do not express in any way their appreciation of the research work before the final deliberation has taken place. I propose now not to delay any further, and therefore, Mr. Van Holm, Wannes, may I invite you to present your research work.
Thank you, Professor Moerik, for the nice introduction. And welcome, everyone. Thank you for being here or listening in online. I hope to both entertain and inform you about what I've been doing in the last 5 years doing my PhD. So, let's start with the oral cavity, also known as the mouth. I see that most of you have one, and that's quite good, because we need it for a variety of functions ranging from respiratory, rhetoric, reproductive, and ruminatory activities. Now, for about half of the people here, what's inside of the oral cavity is especially interesting. Our dentition or in other words, our teeth. Now, for the other half, if you're having any issues with this, you're in quite good company. Today, I don't actually want to focus on our teeth. I want to focus on what's frequently found on top of our teeth, namely the tiny microbes that live with us in the form of biofilms, or collectively known as our microbiome. Now, to understand what I've been working on, I want to introduce you to a few important concepts. The first being symbiosis, in which our microbiome is in balance with their host, and this is health associated, but also quite important in maintaining health because they perform beneficial functions for us. Unfortunately, this balance is not always maintained, and due to a variety of reasons, including poor oral hygiene, this balance can be disrupted in what we call dysbiosis, and this can lead to several oral diseases, and today I'll mainly focus on periodontal disease, also known as gum disease. Well, how does my thesis actually fit in dentistry? Well, the title, Identification and Investigation of Probiotics for Oral Biofilm Management, has an important keyword, being probiotics. What is a probiotic? If you don't know, a probiotic is a live microorganism that we can ingest, and this then in turn confers a health benefit to us, the host. The main question here is which probiotics do we actually consume and which ones are beneficial for us. Second, somewhat related concept is that of prebiotics. Instead of ingesting the beneficials, they're already present in our mouth, and so instead we consume a nutrient that selectively feeds them, and this in turn gives us a health benefit. The main question here is what nutrients do we consume and who do they target? A third, somewhat related concept, or rather a synergy of both of the previous concepts, aims to combine the prebiotic and the probiotic together by designing a prebiotic to stimulate our probiotic. And in this case, our question is, which probiotic do we use and with what do we combine it? Now, with these 5 concepts in mind, what is the goal of my thesis? Well, we're looking at the oral microbiome, and these 3 concepts combined, we can put under the nomer of pro-microbial approaches. And what I want to see is that if these pro-microbial approaches can either prevent dysbiosis or even resolve it by returning back to a symbiotic state, thus oral health. Now, while I believe this is possible, there are several limitations in current probiotic therapy that limit its effectiveness. And addressing these is part of my thesis. Now, we identified 4 key issues, which we then divided into 4 topics, to see if we can unravel and understand the truths behind these issues so that we can potentially improve probiotic therapy in general. Now, let's immediately get started with topic 1, understanding strain specificity. What is strain specificity? It's important to know that not every probiotic will work for a single disease or different diseases. It's not a kind of panacea, and unfortunately also not a panna cotta. Now, you can equate this to how an antibiotic won't cure every type of infection, or an anti-cancer drug won't work on every type of cancer. A quite recent review put it quite succinctly, in which they reviewed different reviews that reviewed probiotics, and they saw that many of these reviews grouped probiotics together without considering any differences in their efficacies between species, and said that any results that these reviews make can be quite misleading or misplaced due to the heterogeneity in probiotic effectiveness. To give you an example of this heterogeneity, let's look at the case of Lactoplantibacillus plantarum. For the non-biologists out here, we classify an organism according to genus, species, and strain information. For us humans, that would be Homo sapiens. And while we don't talk about strains in humans, this information is quite important for probiotics, as the probiotic on your left resulted in improved symptoms of irritable bowel syndrome, while the one on the right, well, technically not a probiotic because it actually worsened everything. Now, even though they have the same species and genus and species identifiers, differences at the strain level resulted in differences in their effectiveness. Now, we believe the same is true for oral probiotics, and together with the University of Antwerp and Unprobiotherapy, we looked at 10 probiotic candidates to see there are differences in their effectiveness and which ones are ideal. Using a variety of methods with differences in complexities, we wanted to tease apart these differences between the strains. And what we saw with the rising of the complexity of these methods is that the differences became increasingly more apparent, with this being the clearest in our biofilm model, where we saw that while All of these candidates were from known probiotic genera, so they were theorized to be effective. They did not share effects and sometimes even completely differed at the species level, and this goes even further at the strain level, so one step down, in which we saw, for example, between these two Reuteris having a completely opposite effect, or for these Streptococcus salivarius strains having more nuanced differences between them. Now, this goes even further if you look at probiotic content. You might hypothesize that having more probiotic will result in more effect. And one of the things we did in this study is correlate these two, and we saw that there was no correlation, indicating that there's actually more an importance behind which probiotic you choose than how much probiotic you have. So, briefly recapping this chapter, using different methods, we were able to tease apart the differences between species and strains, which meant that we saw strain-specific effects, and that which probiotic you chose was paramount of its success. On to topic 2. We looked at that maybe adding a probiotic on its own is insufficient, like how a proper diet for us humans is ideal or important for us to function optimally. We believe the same is true for our tiny microbial friends. Here, the symbiotic concept comes back into play, and using a commonly used oral probiotic, Limosilactobacillus reuteri, together with a quite simple compound, glycerol, we evaluated this concept. Now, how does this work? Well, Reuteris are able to metabolize glycerol like many species, but unlike most, it's able to tolerate and even secrete the normally antimicrobial intermediate, reuterin, and uses to compete against other microorganisms. Now, the sad thing is Reuteris need glycerol, but glycerol is quite limited in our oral cavity, indicating that we maybe need to supplement this. To do this, we did 2 studies in this chapter, or topic, and I'll just highlight some of the most interesting results. From the first study, I want to show you an antagonistic agar experiment in which we spot a pathogen, or rather a pathobiont, next to our probiotic or symbiotic to see its effect on the growth of the pathobiont. Now, in this first image, you can see that the reuteri, so no glycerol added, had a slight inhibitory effect on Aggregatibacter actinomycetum comitans, but did not seem to affect Porphyromonas gingivalis or Prevotella intermedia. The story is completely different for our symbiotic, and this becomes even more clear if we put everything into perspective, seeing that the symbiotic drastically overperformed and even started helping the probiotic out in inhibiting the pathobiont. So the symbiotic had a major improvement in its inhibitory potential of our pathobionts. Now, in the second study, instead of just looking at one strain, we wanted to combine issue 2 with issue 1, in which we wanted to look at strain specificity at the symbiotic effectiveness. And with the help of BioGaia, we looked at the reuteri from before, as well as a bunch of different reuteris in parallel, with and without glycerol. And highlighting the most important figure from this study, we can briefly zoom in to the reuteri we were looking at before. And what we saw is when comparing the probiotic to the 2 controls, they all cluster around 0, indicating no difference between these conditions. So the probiotic didn't do much. But if you look at the symbiotic, there's a drastic decrease in periodontal pathobionts, as well as our anaerobic commensals, indicating its outperformance of the probiotic. We can also zoom back out to the main figure, because we want to compare different reuteris to each other. And what we saw is that many of these reuteris actually had differences in magnitudes on their effects, but they shared the effect, and more importantly, they shared the characteristic that they all needed glycerol to be effective. Without it, they didn't do much. So, briefly recapping these 2 studies together, we saw that the symbiotic always outperformed the probiotic, and strain specificity is also true at the symbiotic level. For topic 3, I want you to briefly think about the microscopic life around us. There's a variety of sources of microbes in our environment that we unavoidably have to enter into our body, and especially our oral cavity, through breathing, drinking, and eating. And most of the time, this is quite harmless. And part of why this is because our innate microbiome is quite resilient to outsiders, and this is a good thing. We want to keep it that way. But you might also suspect that our probiotics are technically also outsiders. So, due to this innate resilience to outsiders, our probiotic can have a hard time of incorporating into our oral surfaces. So, maybe is there a way that we can regain a colonizable surface specifically for our probiotic? Luckily, we have 2 tools in our toolkit, being mouth rinses and toothbrushes. And while today I won't focus too much on toothbrush, if you want, you can scan this QR code to read this article I wrote for EOS that talks a bit about it more, but beware, it's in Dutch. But today I'll mainly focus on mouth rinses. Oh, no, other way. So we looked at a bunch of different mouth rinses, which we naturally combined with our probiotics to see if these probiotics the mouth rinses affected our probiotics, and if we could perhaps combine them together to affect biofilms. Starting with the survival, for once, there was no strain-specific difference. So instead of looking at the strain level, it was more the formulation of the mouth rinse that depended on the killing of the probiotic, so which ones were compatible. Naturally, we also wanted to combine both of them, and when looking at a developed biofilm, where just one of the probiotics had a little bit of incorporation into the biofilm, when we added mouth rinses to the equation, so first mouth rinse and then a probiotic after, we saw 4 different effects. The first being a combined effect, the mouth rinse killing both, so both the oral bacteria as well as our probiotics, not ideal. Perhaps even less ideal is that the mouth rinse didn't do much. Thirdly, already quite interesting, is that while the mouth rinse didn't affect the oral microbiota that much, there was some more probiotic incorporation into the biofilms. And fourthly, the most interesting effect is that there was a combined effect in which both the oral bacteria were reduced, but also more probiotic was incorporating into the biofilm. So, to recap this brief chapter, not every mouth rinse is compatible with our probiotics, and we need to carefully consider which ones you do use. And there might be a thing as stupid as mouth rinse specificity in addition to the probiotic specificity. For my final topic, I want to go back to the definition of a probiotic, a live microorganism. Now, how do you keep something shelf-stable that is alive? Well, we can't really enter a store or a pharmacy to keep refeeding the probiotics because I don't think the police will look very kindly upon us. So are there maybe ways that we can circumvent this? And yes, indeed. One of the techniques we use for this is lyophilization, or in other words, freeze drying. And before I explain how this works, I want you to tell me what this is. Okay, I hear yeast. Some bakers in the audience. It is indeed yeast. And now collectively point either to your left or your right. No, your left or your right. Which one has the longest shelf life of both of them? Yes, most of you are pointing left, and that is indeed dried yeast. Why does it have a longer shelf life? Well, we just remove the water. Many microorganisms, including our probiotics, have the ability to enter a kind of hibernating state, if done correctly, by removing water so that they can be stored for a bit longer before they are rehydrated and resume their activity. One of the ways that we can do this is through lyophilization, in which we freeze water into ice as rapidly as possible, then lower the pressure, and when we reheat the samples, instead of the ice turning back into water, it immediately evaporates into steam, and if we use a condenser to remove it from the product, we get a quite stable dried product. Now, the issue here is that our probiotics don't necessarily survive this, and this led us to see if maybe looking at products on the market, if you could see what they actually contained and how much of the probiotic they should contain is actually still alive. So, when analyzing what we saw is that the promised amount of probiotics that they were going to deliver was in a somewhat similar range, but when actually looking at the total living and culturable cells, we saw that there was a lot of heterogeneity and a lot of differences in quality between these products. This is quite sad that so many of these contain so many dead probiotics, leading us to, of course, ask, why does this happen? To just give you a very brief overview of why this could happen is that there are 2 things that could mainly go wrong. Either during the preparation, so the lyophilization, if it's performed improperly, our cells will immediately be damaged, resulting in their very rapid death, or like any product, if it's stored at an incorrect temperature, if it doesn't have any preservatives or whatever, there will be a gradual decay in these products, and it's even worse for the probiotic because before the product actually goes off, the probiotic might have already died. Now, this can lead us to ask a variety of questions about probiotic efficacy, but what we focused on most was adhesive potential. For a probiotic to be an oral probiotic, we want it to adhere in our oral tissues. If it doesn't, it gets washed down to our gut, making it more a gut probiotic than an oral probiotic. So one of the things we tested is looking at the adhesive potential of a fresh probiotic against a dried probiotic. And what we saw is that the fresh one always outperformed our dried probiotic. And this is quite sad, because most of the probiotic products on the market are in a dried formulation. Now, maybe we can also regain this adhesive potential. So that's what we did next. And by comparing a fresh versus a dried probiotic, as well as reactivated or rehydrated probiotics for 24 or even 2 hours, resulted in most of this adhesive potential being regained. And this might be interesting for future product development. Now, briefly recapping this project as well. Current product preparation might result in a lot of dead probiotics, unfortunately, that don't really want to stick around in our oral cavity. And I think we need to maybe look at this. Now, I've also actually reached the end of my short presentation. But before I let you ask some questions, I want to give you some key takeaway messages, about what I've discussed, so that you can remember it, hopefully, until your deathbed. Yeah, a bit mortifying, sorry. So, first takeaway, strain specificity. We need to carefully consider which probiotic you use, as this can determine the efficacy of probiotic therapy in general. Secondly, symbiotics over probiotics. We need to consider the diet of our tiny friends for them to be more effective. And once again, strain specificity for probiotics as well, symbiotics as well. And then thirdly, if we want to combine antiseptics or mouth rinses together with probiotics, we need to carefully consider which are combinable, because this can have a big effect on the ability of the probiotic to colonize our oral cavity. And then fourthly, current product preparation of oral probiotics might result in a lot of dead probiotics, and the ones that are still alive might not necessarily want to stick around and become maybe more gut probiotics. And we probably should address this in future to improve probiotic delivery and thus probiotic therapy in general. And that was all. Thank you very much for listening, and I look forward to your questions.
Hello? Yeah. Okay, it works. Thank you, Ms. Van Holm, Wannes, for your presentation. You asked us in the audience already some questions, but now it's the turn of the examining committee to ask you some questions. And I suggest we have a quite nice group of jury members, 5 in total, and we are pleased that they all are present here today. And therefore, I suggest that we limit to 2 main questions, let's say, but of course they can also go into a little discussion with you and challenge you further if they want to. So may I first invite Dr. Mira to examine the candidate.
Thank you very much, Mr. President. Thank you very much, Mr. Van Holm, for the invitation to be here. It's a very big honor. I have been asked not to express any appreciation of the research during my questions, and it is a pity because I have liked to express at least the amount of work that is involved. I can assure you that I am aware of the amount of work involved in this thesis and the relevance it has. I have a first question in relation to your the model that you are using, the biofilm model where you select a number of species. I would like to know, in comparison to other models where people take real samples from the patient and then culture them, in that case you have a biofilm with over 100 species that resembles more the real sample, but of course that varies more between individuals and it's more difficult to standardize. So I would like to know your view about the use of your model where you select those species in comparison to other models where people use real samples in a microcosm model. what do you think are the pros and cons of them?
Yeah, absolutely. Thank you for that very acute question. And this is indeed something that we are struggling, or well, not struggling, but debating all of the time. So we use this model for a while now, and the nice thing that is quite repeatable, and that's one of the main issues, main benefits of this model, that we keep reusing this. But indeed, it misses a lot of genera and species that are actually quite clinically relevant, and models such as that you're working on, but we are also working on an ex vivo model to balance both of these. So ideally, if we have the correct expertise, through sequencing everything, we try to balance both of them by combining them and looking at them together. But yeah, the main issue with ex vivo or samples from patients is that even if you take from the same patient, it won't be the same every time, so resulting in the repeatability of that setup being somewhat limited, but more clinically relevant for at least that patient, and our model being somewhat more repeatable, but less clinically relevant. Mm-hmm. Ideally, there can be worked towards a kind of shared like a functional ecosystem that I'm forgetting the word that I used, a core microbiome, that they share different functions, that even though there is differences between the species, as least as the same type is included in your model, it will result in a repeatable experiment, or at least, depending on your treatment that you do, that it's repeatable despite having differences between patients or samples.
Okay. In relation to that, I saw that in the model, in the control, some of the organisms that were, I don't know, 2 or 3 species accounted for a big portion of the total. Is it because you actually add them in those levels, or you add them all the different species at the same proportions, and some of them happen to grow more than others under those conditions?
We add them at approximately the same cell densities into the bioreactor, and then after it's stabilized, some of the species become more abundant. Villanella parvula is one of the most abundant ones, okay, which we'll probably have a discussion about as well. But okay, indeed, there are more abundant species, and depending on also which kind of parameters that we have during the biofilm formation, we can modulate the biofilm into directions being more symbiotic or more dysbiotic, and I think Nayara's paper is almost published about this as well. Mm-hmm.
Okay, my second question is in relation to the use. I mean, the species that you have, the strains that you have. That you have selected as probiotics to test. Some of them are of oral origin, but mainly you have selected species from the gut or even from other human niches. At the beginning, when the first tests were done in the oral cavity, normally gut species were used, and they were not able to colonize very well, and the results were not great, and people thought, well, perhaps it's because they are non-oral. And I think many people were waiting for our work and others looking at Esdentisani and other species which are oral to see whether they were able to colonize. But then we saw that the same, they are able to colonize for 2-3 weeks, and after 1 month, they seem to fade away. So now I'm not sure anymore. I would like to know, what do you think? Do you think that we should use oral species as oral probiotics, or it doesn't matter? What was your thoughts about that?
So I think we definitely need to have species that are able to form biofilms and interactions with communities. If these interactions have to be already designed to oral cavity, depends. I think especially choosing or selecting them from healthy individuals might already give us a head start on species that we know that can colonize, but maybe sometimes we need to introduce a wild card that has, in a completely different ecosystem, a very interesting effect that we don't normally have in our oral microbiome, because many of these beneficial species that we isolate are maybe more important in maintaining oral health, but maybe we need to find, like, well, not a nuke, but something that can drastically shift it towards health back again, and then maybe introduce maintaining species such that are already found in healthy individuals. So maybe there are 2 phases of probiotic therapy, a more intense one and a more maintained one. Mm-hmm. That is my opinion.
Okay. I will finish by a short question from an applied point of view. You have very clearly shown that the fresh isolates always outcompeted the powder, the lyophilized in terms of being able to attach better and so on. and also that it takes a few hours. Actually, I think you showed that 24 hours is when you get the maximum, right?
Yes.
And that's never going to be achieved because you would swallow that in the saliva and it would never be able to exert its function. So how do you see that solved in practical terms? You talked about having this reactivation. How do you think that could be solved in a real product?
Yeah, Wim won't like that I say this, but there are certain formulations that I do like, and there are a few articles about this as well, like yogurts. Maybe we design yogurts that have the correct probiotics as well. I don't know if they'll taste great, but then they hopefully stay alive a little bit longer because they can contain active cultures. One of the things I'm also quite interested in, and one of Nico's ex-students is working a lot on kombuchas, which are also that you can maintain your SCOBY with your probiotics, but this is maybe already a lot more work. But I think for actual product preparation, I think that like making a flavored drink together with the probiotic that you, for example, you're making your breakfast and you add your probiotic powder and a flavor agent to your bottle, shake well, and you keep it until you go to work.
And wait for a while.
Yeah, and then once you have given it 2 hours to activate and start regaining its adhesive potentials, if you slowly start sipping it, I hope most of the probiotic will be delivered. But that's
so you're mainly thinking about the product being used at home. I mean, do you also see that being used at the clinic, and perhaps they have more time to prepare it before the visit of the patient?
That is also, I think. But there the legislation comes in. So having a live culture into something that's a medical device is tricky. If it's performed correctly, I think this could be also a good way, that having a kind of slower release of it, that you can introduce it instead of a lozenge or a tablet, finding alternatives to let them slower release and not being washed away could be definitely an improvement. Also timing of when you take the probiotic is quite important. If you're swallowing a lot of saliva, not really. If you're just laying there and sleeping, less saliva flow and perhaps more probiotic adhesion.
Okay, thank you very much for your answers.
Thank you, Dr. Mira. I will hand over the microphone now to Professor Kroon.
Thank you. I've looked forward for a while to exchange my thoughts with you, and I have a lot of questions, but I will focus on some of my key aspects. My first question is kind of continuing on something that my colleague Dr. Mira started, and that's about your observation that there's a species or even a strain-specific activity of your probiotics. And I think that you're right, and I think that that's very important, but that this strain specificity, do you think that that also holds true for your synthetic community that you're using?
Definitely, yes. So we have, or in literature, there are different cases of periodontitis that don't necessarily rely that much on the species that we include. So it's more community effect that...
But also within strains, right? I mean, within AA, you have certain strains that are associated with the aggressive form and some that are not. Yep. So the strain level in your synthetic community, I guess, is also of importance. Definitely.
So we try to select pathogenic strains that are also in type collections, but I definitely think we also have a bunch of Porphyromonas with different capsule types, and we know that they react completely different, and we actually should test this as well. But then by changing this, the model also completely changes, and it's definitely something that should be tested, but requires a lot of work and maybe some extra hands in the lab. Yeah.
But so it's very well possible that if you look at your pathobionts, different strains of your pathobionts, you might find other probiotics that might be active, while the ones that are in your synthetic community that are active against it, they might not be.
Absolutely.
So you have kind of a microbiota-specific probiotic.
Yeah,
so let's make it a bit more complicated even.
Okay, sure.
Let's involve the host, even though I know that we don't have strains of people, but we are very variable. And of course, we are very different. We do have a core microbiota in species, but everyone has their own strains, which you probably in some way get from your mother, or there's some way in which your mother or your parents kind of allow you to get certain strains and others do not. So if you have strain-specific probiotics, you have strain-specific oral microbiota and pathogens, and now you also have a host that might vary, would that mean that then you'll have a personalized probiotic that we need, so where there's 300 on the shelf and you get a type A, B probiotic type?
Yes, I think personalized medicine is definitely for like these microbiome-related diseases. If it's not a single pathogen, but more an interplay between the microbiome, the host, and the environment, we need to really identify what is important for one person. But this needs a lot of big data analysis that we need to both characterize the host's response and everything. And I think, especially for probiotics, if they want to interact with the host, there's a lot of immune reactions that can completely differ between probiotics. And your probiotic that maybe works for microbiome modulation maybe doesn't necessarily work for our immunomodulation. So maybe we need to combine different probiotics and create probiotic cocktails that can thus be tailored to a person for their specific needs. But I think that's big data.
Tailored for a person during a specific period in their lifetime, because we change over our lifetime. So yeah, it gets very complicated, but it's very interesting. So I have a very short question in between. I read in the introduction a very beautiful sentence, and it reads, "Tucked away within the diverse biofilms, patiently waiting for their opportunity, hide naturally present pathobionts." So if you're not successful in this defense, I think you can become a writer of novels. It's a beautiful sentence, one of the best that I've ever seen. But I want to challenge you on this concept. Because why do you think that they're patiently waiting in order to become a pathobiont?
I have how do you say it, the beroepsmisvorming, that I start attributing personalities to the bacteria that we work with. And so after a while, I start thinking, oh, they're out there sneakily, so like a small trickster. But I don't think they are actively doing this on purpose. They see an opportunity, and like any of us, we would grab an opportunity if we don't think it immediately harms everyone else. Yeah. Like, especially with outgrowth diseases, if we look at Candida, if everything is removed, the community control around them falls away. And they said, yeah, there's nobody stopping me. I can just keep growing. So it's rather not focused on being harmful, but more opportunistic. Yeah.
Well, thank you for mentioning my pet organism. So now I can continue on that. The idea that you have these pathogens that are present in a commensal system, because PG and AA, all of them are present at very low levels also in health. Mm-hmm. Do you think, and would you agree with me, and one of my hypotheses, and I hope in 15 years I can have sufficient support for that, is that these opportunistic pathogens in commensal systems have a critical role, otherwise we would not allow them to be there. Because we do not allow Pseudomonas to be in our oral cavity, because it's a true pathogen. Mm-hmm. So would you follow me in saying that these pathobionts that are lurking around and waiting to become pathogenic might also have a positive role in the commensal microenvironment? And that is why we allow them to be in our oral cavity?
I definitely believe this could be the case. It's quite maybe controversial to say in a way, but like in the case of caries, if we want to balance our periopathogens against the caryopathogens, they're in direct antagonism. So if you would have an excessive amount of sugar intake, maybe there's a possibility that the proteolytic periodontal bacteria are able to suppress outgrowth of our karyogenic bacteria. So in certain circumstances, I think they can give an important counter to other developing dysbioses. But yeah, the evidence on this is maybe still lacking behind because of the thoughts of the red complex and not the polymicrobial synergy.
Well, I think that as far as I'm concerned, the pH effect, that might be something that might be of interest, because of course the periopathogens, they have these higher pH, and the karyogenic pathogens, they have the low pH. Do you have time for a small question? Or? There was one thing that I found remarkable, and that is in your chapter 3. You look at the response of keratinocytes to LPS, and then also in relation to your probiotics. And then you state somewhere that keratinocytes didn't give a lot of response, especially not for IL-8, I think, and maybe TNF-alpha, that it was a low response. And then you say, then we chose to use THP1 monocytes. Why did you not use a fibroblast cell line?
So the thing is that the keratinocytes did produce IL-8, but due to the presence of, because we tried it first with biofilms, and Porphyromonas gingivalis has the nice habit of breaking down IL-8. So having Porphyromonas together with that resulted in very low IL-8 production. We also tried mimicking or working towards the Scheim reactor from Professor Don van der Wielen, but instead with working the keratinocytes, fibroblasts, and THP1s to also create like a structure. It's not that advanced yet, but we compared them as well. But the most potent producers, especially for IL-10, was the THP1s. And so, yes, we could also check the fibroblasts, but I had to choose which ones I went. And there were a bunch of articles that already used just LPS, so no PG itself, but the LPS of PG, together with the THP1s and also probiotics. So I inspired myself from previous literature building on those, but I think fibroblasts would definitely work as well.
Yeah. Well, I think that they're definitely one of the cell lines that would be my first choice, because the keratinocytes are your barrier, and they're used to seeing the microbes. And then and your fibroblasts, once the fibroblasts start to see them, then your barrier has been broken. And so they respond, in our models at least, the fibroblasts, they are 10, 20 times higher in their response to any stimulus that we give. Yeah. Thank you very much.
Naira, are you writing this down?
Thank you. Professor Van der Wielen.
Thank you very much, Mr. Chairman. The candidates, thanks for sharing your work with us, very interesting. I would like to continue the discussion that you just had with Professor Krom regarding the pathobionts, because he asked a very smart question regarding the role that these pathobionts may have within the commensal microbiome as such. I would like to take it one step further. And is there a certain role of pathobionts also for the host directly? And how do you see this? And maybe you can think of how literature evolved starting from this concept of pathobionts, because that was a little bit the terminology critical questions that I had in the internal review.
That is a very philosophical question. I definitely think there's like a co-evolution between our commensals, our pathobionts, and everything that goes throughout time. Our new model is moving away from a causal pathogen, but more a balance between host and microbiome. And one of the, I don't know the exact quote, but one of the quotes of saying our current view is that there's more an intricate play between the host and the microbiome at an inflammatory level. So there's always a very low basal state of inflammation. So if there's no inflammation or way too much, you get completely different pathologies, but there's always a basic layer of a small like war between our pathobionts and us, or our microbiome in general and us. And I think, like looking at Alice in Wonderland, we have the Red Queen hypothesis. There's a constant fight between them, evolving towards how humans is. Unfortunately, I don't think we have a lot of data at looking at the oral microbiome throughout time, how this changed, but yeah, if ever we find Neanderthals that still have an intact microbiome, maybe we could see something very interesting from this. I hope this answered your question a bit.
Yes, it certainly answered the questions, because you mentioned, okay, you need a certain grade of inflammation to that facilitates also that co-evolution. Maybe we should not even call it a war, but even more like a sparring partner or something. Yeah. So in that sense, I think I was wondering whether you could give a couple of examples, but I do acknowledge that most of the examples that we know is probably more coming from the gut environment. But I'm not sure whether you could give an example.
Yeah, I think, I don't know for the oral cavity that much, maybe I should look into it more, but like especially from virology, we have the co-evolution from a new virus versus a virus that has been with us for a long time. The difference in how the host responds to a new virus, especially with he shall not be named from a few years ago, which some young people that have never been in contact with a COVID virus, oh, I said it, very extremely respond, and their immune system actually just goes crazy. And then you have the other viruses that have been with a long time with us, which just make us a little bit sick, but it's more a balance between not really harming the host and staying alive, and going further. And I think the same, in some way, of this coevolution, but over a longer time period, happened with our oral microbiome.
Yeah, that's true, because you can actually even trace human beings and their migration across the planet also together with their microbes. And,
Yes.
and many of these are actual pathogens or opportunistic pathogens.
Indeed, spatiotemporal instead of actual temporal throughout history, analyzing the differences, yes.
Yeah. Maybe a little bit more of a methodological question. Of course, you have this biofilm model that is very attractive to test all kinds of hypotheses regarding pure cultures, specific microbes, pathogens, probiotics, multi-compositions, consortia, and so on. This relies a lot on the hydroxyapatite discs, but of course, the actual environment that we are having in the oral cavity regarding the adhesion to the tooth surface is still a little bit different. Could you enlighten us a little bit how this works?
So we also have a very nice model of someone we'll have to defend quite soon that uses a more dynamic. So especially what's wrong with our model, the hydroxyapatite doesn't have the same same characteristics as dental enamel. There are different ones that are mimicking closer, but I think the most important difference, rather than the hydroxyapatite, is the dynamic nature of our model. We are using it quite statically, while the oral cavity is continuously refreshing, removing stuff, adding new nutrients. And I think this is one of the things that we need to improve on the most, to actually mimic more the oral cavity.
Yeah. So that's still, I completely agree, the dynamics need to be certainly covered. There's also this little layer that is in between still, so the acquired animal pellicle. Is there any functional role there? I mean, would we substantially see a different colonization potential from our different microbes under study if we tune in towards that pellicle or not?
So we try to use other mucins, so maybe not the nicest thing is using porcine mucin from the more gut mucin,
it's
but we believe
commercially. So
yes, and harvesting saliva works for some setups, but it gets really tiring after a while. But we believe that the salivary mucins behave relatively the same as the porcine mucins, but I think moving on to clinical would be ideal. But effects of the pellicle and how it interacts can hopefully, or might differ, but I don't know how much.
Okay, good. Thank you very much.
Okay.
Thank you. I give now the word to Professor Van Holm.
Wannes, it is indeed a lot of work, so I had some time to go through, but I enjoy it.
I hope you still have some questions remaining.
Yes, I think so. One of my questions is about antagonistic tests you did. You used the antagonistic agar assay, and also you measured it by flow cytometry.
Yes.
But for the flow cytometry, you used cell-free supernatants from your probiotics, while for the agar test, you used real bacteria. Yeah, okay. And then you come to the conclusion it is something, discrepancy that I noticed. My question is, why didn't you do the flow cytometry with more than one cell? Because I remember that you said that you could divide different shapes of bacteria in a flow cytometer analysis. And could it be then not give more information? And vice versa, could you put your cell-free supernatants on an agar and see what is happening with your cells?
So I'll first answer for the flow cytometry and then go to the cells. Yes, we can indeed divide cells or look at them separately, but there's some overlap between the gates. And Fabian, who's working on this, is using a random forest model to be able to separate them properly. And while we could do this, the clarity of the signal became a lot more muddled because the overlap of the live and the dead gates, when we used both the probiotic and the pathogen, resulted in us mostly seeing the probiotic. So even though the pathogen might have been killed, the amount of probiotics remains a constant. So seeing a reduction, we would have to dilute our sample further, and it wouldn't matter because we can't dilute if the probiotic is still there. So if a pathobiont is killed, we use a lower dilution. But if we dilute lower, the flow cytometer gets overloaded because the probiotic is still very live. So that's the reason why we opted for the cell-free supernatant, so the postbiotic more, to have a clearer signal. As for the, I hope this is already okay for the flow cytometry. And then for the agar experiments, I did this indeed, that spotting the cell-free supernatant on top of it kills it, or might kill it, but afterwards it can start growing again. So
what was the last? It will. Yeah, you say.
I think it should be able to kill it, but after a while they still start growing. So sometimes with Rita we tried this a little bit, that we saw that the morphology of the colony changed, so that instead of having a uniform ecology, we had more spotted, but after a while these spots started going out, resulting in still the same diameter, and also looking at the distance and everything, it became hard to get something quite uniform as the flow cytometry.
Okay, so in your work there is a lot said about bacteriocins, yes. So, and one of your remarks is you say, for example, salivarisin B has no antibacterial activity against gram-negatives. This might explain the effect of salivarius strains on commensal streptococci. But streptococci are gram-positive, so why is your bacteriocin or your compound not working against streptococci? Why is, or I misunderstood, that you said that salivarisin B has no activity against gram-negatives. Okay, I agree. But this might explain the effect on of the salivarius strains on commensal streptococci.
So there was a slight reduction of the commensal streptococci.
Is it?
Yeah, very slight. Not that much.
But I thought it was more an increase, but okay. Yeah, okay. I didn't see that decrease, I will say it in this way.
That I should check again,
yeah,
but
okay. Yeah,
no,
okay.
that might be a discrepancy indeed. That, yeah,
but
I'll have to double check it.
I think you were explaining this because you said, I think that the hydrogen peroxide will be more important than the salivarisin B. I think that is. But do you have an idea if those Streptococcus salivarius is producing salivarisin B or A or whatever?
So
did
for
you
that,
check the gene, or did you check if there was something bacteriocin-like thing?
I don't know from these, but I have someone in the audience that maybe does know, Tom. No, but so they have the whole sequences. I think they also maybe have the if the patterns are still remained, or like the genes are there. I'm not 100% sure, but I didn't know
because
it for
a lot
these.
of strains
Don't necessarily,
don't have them, so
or produce very little.
Yeah, it was a little.
Yeah.
I found it a quick thing. And also you said for the Lactobacillus reuteri, I don't know what is the real name of the L reuteri, that there was a paper about a proteinase reuterin, and then you said, yes, probably there are other, how you say it, inhibitory mechanisms.
Yeah.
Can the compound be a bacteriocin-like thing?
Possibly. So
because it was 52 kilodalton, the protein which was produced there.
It is, I think 52 kilodalton is quite big for a bacteriocin. I think they're usually smaller, but maybe Professor Van Wille knows better.
What are bacteriocins?
Yeah,
how do you describe?
It's actually probably also a way too big term. So there are many types of bacteriocins, ranging from smaller compounds to bigger compounds. Yeah. But I'm not the expert in antimicrobial.
But it is really a large amount of bacteriocins.
It's a big number for probably way too much.
Okay. A second thing I was thinking about when I was reading, you are using glycerol a lot, and sometimes you use 200 millimolar, then you use 2 molar glycerol. For example, when I I use 20% glycerol to preserve my stocks. And you are saying also in the toothpastes and so on, a lot of glycerol, varying concentrations. What about, for example, the 2 molar concentration? Is this clinical relevant? Is this comparable with toothpastes? What is the concentration in toothpastes? Why did you choose those concentrations?
Okay, so 2 molar is actually 7%, or 14%. 14% glycerol, and in several mouth rinses, especially Xeros, it has about, I think, even 30% of glycerol because it helps with feeling of dry mouth. So definitely it's a concentration that can be used. If it's a good concentration, I'm not 100% sure because I think these concentrations can also affect, if you have too much glycerol, it can also affect the activity of the reuteri, because this kind of concentrations also start affecting the water activity. So
right, the water activity.
Yes, yes, indeed. And
so can you explain what it is exactly is, the water activity? You're mentioning that water activity of 10 millimolar glycerol can already be detrimental for bacteria. So, and I thought he is using so much. So, what is that water activity then?
I prefer to actually go to lower glycerol concentrations, but at these concentrations, how does it affect it again? I should have reread this because it's been a few years, and I don't think the professors from molecular or of molecular biology. But indeed,
I
it
was
affects
curious about to read it, so therefore I.
But I have to admit that I've.
No. So, and can you compare the water activity of glycerol with sucrose or skim milk, or is this a completely different system, or can you compare those? I'm using glycerol for my stocks. You were using skim milk and sucrose. Is that the same mechanism that we are using there, or?
I think the concentrations of the skim milk and the sucrose are a bit lower. Also, that the way they dilute is slightly different. So in a much larger volume. But
also something to do with the water activity.
That they inhibit or protect. For creating the product. No, so the skim milk has a lot of different compounds that either can protect our cell wall and cell membranes. For the sucrose, how does it work again as well? As lyoprotectants, it stabilizes the membrane in different ways, and I think it depends also on the compound, especially for the cryotherapy. The glycerol or freezing something, the glycerol stops the large crystals of forming that completely puncture our cells and rip them apart. As for sucrose, I know trehalose is very important because I did my master thesis on this as a cryoprotectant, but how it actually works. Oof.
Okay, we will look it up. Thank you.
Thank you.
Thank you, and then I give the microphone to Professor Michiels.
Thank you, Mr. Chairman. So being an assessor of you, I've had the opportunity to discuss your work into more detail over the past years. So I will limit myself to 2 very general questions. And the first one is, so you indicate in your literature that depending on the study, the oral bacterium consists of about 700 to 1,000 species with maybe 50% unculturable or unknown. This is a huge number, no? Do we all have that number of species? And so what's the variability? A lot is known on the gut microbiome. Is similar information available for the oral microbiome? Are there differences between people, between geographical origins? And is there some kind of a functional meaning of differences?
Yes, so hopefully I remembered every single one of these, but of the 700, we don't typically have all of them. Usually our estimates are that we might have around up to 150 of this collection, and depending on the sites, it maybe ranges up to 50 species. I think it's also important to note that maybe while having different species, they can form or perform the same functions in different things. And I think these differences can also differ between populations on how they respond to different treatments. I know for host modulators, there's a lot of differences in how they respond depending on, but that can maybe be the genetics of the human itself, as well as the differences in the microbiome. I think there's a lot of bias in literature towards more Western research, but I think there was a Nature article published last year about also the differences between in families, larger groups, and big ones. I can't remember the name by heart, but it really accurately describes spatial-temporal differences. But I don't think necessarily the response of the humans to treatment. Did I forget maybe something of your questions?
No, is there something functionally known about differences in composition related to, I don't know, function? Does it have a function maybe first? And
so in health, I'm not 100% sure, but in developing of a disease, definitely yes, because the composition starts to shift and also becomes to diversify or shift as well. But between populations, I'm not 100% sure, but I believe that there's definitely differences that could attribute to some people developing more certain diseases, like especially now maybe in the States, dental decay might be a lot more due to the increased carbohydrate consumption, resulting in their microbiome shifting as a population towards a more carcinogenic, or maybe people who eat way less refined sugars will have a different kind of microbiome, resulting in less caries, but maybe more periodontal disease. But I have to check literature again to really give you some good sources.
okay. And secondly then, let's hypothesize that you will have your PhD degree by the end of the evening, and that your promoter is so happy that he gives you a lot of money. The one being more likely than the other, I guess. So if you would, based on your results here, if you would continue your work, what direction would you go? What would be the perspective and the aim of your research?
Okay. I'll take advice from the commission of my final doctoral plan and try to limit my answers, that I don't have too many side projects. But the most things I want to focus is on the actual retainment of our probiotics in our oral cavity. I believe that many probiotics could be functional, but if they don't want to be retained, they won't be functional. So looking at better delivery methods, slow release devices, alternate deliveries, delivering something fresh versus inactive, that's something I would like to work on. Also seeing if we can maybe understand more the different responses of people to different probiotics, or like if you have different core periodontal microbiomes, how do they respond to a certain probiotic? Are there patient differences, and do we need to work towards understanding this? That'll be a very big task, in which we will need to hire someone who's way better at bioinformatics than me. And thirdly, I also would want to look at more the microbiome in general, not just bacteria, looking at fungi, viruses as well, how they interact, because I think there are so many interactions that we're just ignoring, which we shouldn't. So those three things, and I'll hide the other 10 things in the drawer somewhere.
Okay, thank you.
Thank you, Professor Michiels. It's not over yet. I have 3 promoters here, and I expect that they will ask you, this is the final time you can, some further questions. Professor Teugels, Boon, Bernards. Okay. The audience, is there somebody in the audience that wants to ask a question? It doesn't happen that many times, but okay, if not, then thank you. I would like to invite the members of the examining committee to follow me for the final deliberation. On behalf and under the authority of Professor Luc Sels, Rector of KU Leuven, and Professor Rick van de Walle, Rector of UGent, considering that Wannes Van Holm, born in Leuven on April 12, 1993, is a holder of the Diploma of Master of Biology issued by KU Leuven in 2018, seeing the statement of the examining board appointed by the rector, from which it appears that Wannes Van Holm has submitted and publicly defended a thesis on identification and investigation of probiotics for oral biofilm management. On the advice of the examining board and in accordance with the requirements of the decrees concerning higher education in Flanders, I hereby confirm upon Wannes Van Holm the degree of Doctor of Biomedical Sciences at KU Leuven and also Doctor of Applied Biological Sciences at UGent. Congratulations. There is one final remark or a message that I have to give you. By obtaining the doctoral degree, you commit yourself as a doctor to act in the future in accordance with the basic principles of research integrity as described in the ALEA European Code of Conduct, reliability, honesty, respect, and accountability. Please be seated, and I think that I now want to give the word to the promoter.
Ja, daar blijven staan. Ja, daar blijven oefenen voor blijven. Is oké. Goed. Dames en heren, geachte gasten, vrienden, familie. Vandaag komen we hier samen om een opmerkelijke prestatie te vieren. Een prestatie die staat voor jaren van hard werken, toewijding en meedogenloos streven naar kennis. Het is mij dan ook een grote eer om voor u te staan, om de opmerkelijke reis te vieren die dokter Wannes Van Holm heeft gemaakt en die uiteindelijk heeft geleid tot de hoogste academische graad aan de universiteit als dokter in de biomedische wetenschap. Een doctoraat behalen is geen eenvoudige opgave. Het is een bewijs van iemands streven naar uitmuntendheid. Iemands honger naar kennis en een niet aflatende vastberadenheid om bij te dragen, niet alleen aan onze wereld hier, maar aan de wereld in zijn geheel. En dit door middel van iets wat we noemen baanbrekend onderzoek en ontdekkingen. En vandaag erkennen we de ongelooflijke reis die Wannes is begonnen. Een reis die heeft geleid tot deze belangrijke mijlpaal in zijn leven. De toewijding van Wannes Van Holm aan ons vakgebied, de orale microbiologie, is ontzagwekkend. Zijn passie voor leren, zijn intellectuele nieuwsgierigheid en zijn onvermoeibare zoektocht naar antwoorden. En ik moet heel eerlijk zijn, voor hem is het misschien onvermoeibaar, maar voor ons als promotoren was het vaak heel vermoeiend. Maar bon, hebben allemaal een cruciale rol gespeeld in het bereiken van dit buitengewone moment. Het nastreven van een doctoraat vereist talloze uren van onderzoek en experimenteren, zoals je wel weet, Wannes. En Wannes heeft dit uitdagende pad met gratie en vastberadenheid gevolgd. Wannes, je toewijding aan de orale microbiologie is werkelijk indrukwekkend. En initieel dachten we allemaal dat je dit alleen maar deed om indruk te maken op je kat met al die nachtelijke onderzoeksuren, maar kijk waar het je uiteindelijk heeft gebracht. Maar naast je academische verwezenlijkingen was er blijkbaar nog tijd over. Had ik dat geweten, had ik andere zaken op je boterham gestuurd. Maar er was tijd, want op de meest onverwachte en vaak onmogelijke momenten in mijn eigen agenda kwam je met specialekes af. En ik denk hier heel specifiek aan je ontwerp van het logo van de onderzoeksgroep. Dat is een van die specialekes geweest. Het inkleuren, zoals je kan allemaal zien op zijn thesis, het inkleuren van SEM-beelden compleet nieuw was voor ons in onze onderzoeksgroep. En je bent eigenlijk zelfs zo ver gegaan dat je een telefoonhoesje hebt gemaakt in de vorm van een orale biofilm. En met dat telefoonhoesje heb je zelfs een Europese prijs gewonnen. Dus voor degenen die geïnteresseerd zijn, als je voor 23 uur bestelt op bol.com, dan heb je morgen in de bus. Wannes, met deze prestatie sluit je je aan bij een heel select groepje van wetenschappers die blijk hebben gegeven van een uitzonderlijke inzet voor het streven naar kennis. En je behoort nu tot die experts en pioniers op jouw vakgebied en jouw werk zal ongetwijfeld een blijvende impact hebben op de wereld. Ik zag achteraan in jouw thesis staan, terwijl je hier aan het verdedigen was. Het was een beetje saai voor mij, dus ik ben eens gaan kijken en ik zag daar iets staan van een mediocre researcher. Ik denk dat je jezelf niet als mediocre mocht zien. Je onderzoek heeft de grenzen van ons begrip verlegd en uw bijdragen zullen in de toekomst onze discipline zeker nog verder gaan vormgeven. Ze zeggen, Wannes, dat een doctoraat als een huwelijk is. Het kost tijd, geduld en een gevoel voor humor. En geloof me, nu weet je al dat het niet gemakkelijk is om getrouwd te zijn. Maar Wannes, het doctoraat is er al en je lacht nog steeds, dus waarschijnlijk gaat dat huwelijk ook wel goed komen op een gegeven moment. We moeten ook erkennen dat een PhD niet alleen maar gaat over artikels en experimenten. Het gaat eigenlijk over overleven. In Wannes zijn geval 5 jaar overleven bij ons op de onderzoeksgroep. En dit kan je duidelijk, dat heb ik nu van jou geleerd, op een dieet van iets wat we noemen zwart water van op het 5e verdiep, dat jij koffie noemt, en proteïneshakes. Ik bedoel, wie heeft er dan nog een doctoraat nodig als je alleen daarop kan overleven, Wannes? Ik weet eigenlijk niet waarom je dat doctoraat gedaan hebt. Ik had eigenlijk willen zeggen dat jouw bijdrage aan de orale microbiologie is als een gastronomische maaltijd voor de hersenen. Het is heerlijk en bevredigend. Maar als ik dan denk aan dat zwarte water en die proteïneshakes, dan weet ik eigenlijk niet zo goed of dat de juiste uitspraak is in jouw geval. Dit is niet alleen een moment om te vieren, maar ook een moment om dankbaar te zijn. Wannes heeft deze mijlpaal niet alleen bereikt. Zijn reis werd ondersteund door niet stoppende aanmoedigingen van zekere vrienden en familie, van de begeleiding van de mentoren en ook van collectieve inspanningen van collega's. Nee, Wannes, je bijdrage tijdens de afgelopen 5 jaren waren allesbehalve beperkt tot je academische verwezenlijkingen. Wat mij vooral bijgebleven is, Wannes, is je rol als lijm tussen de mensen, mijn groep op het labo en de klinische assistenten. Het is mij zeer duidelijk geworden dat je daar een heel belangrijke rol hebt gespeeld en ik hoop nog zult blijven spelen in de komende jaren, dat je hopelijk nog wat bij mij blijft. En het is mij ook duidelijk geworden dat de assistenten dat appreciëren of geapprecieerd hebben. Anders zouden ze niet met jou je outfit voor vandaag zijn gaan kiezen. En gelukkig maar, want anders stond je hier vandaag waarschijnlijk in korte broek. Bon. Ik wil ook van de gelegenheid gebruik maken om de leden van de jury van ganser harte te bedanken. Dear Alex, dear Bastiaan, dear Lieve, Tom, and Jan. I often hear people around me say that we have a luxurious life. We only have to teach 10 hours a week. We have about 3 to 4 months of vacation. We are traveling all over the world. And we all know well that this is not true. We certainly have a full life. I'm not going to challenge that. But working days of 12 hours and more are more the rule than the exception, as you all know. In addition to that all, we still have the stress of fundings, problems with students, not with Wannes, nevertheless, and so on. And you also have these obligations, these colleagues, these friends that ask you to be a jury member for a PhD. And people here might think, how horrible can that be to be a jury member? You have to sit there for 2 hours in a gown, go for a good dinner afterwards, and that's it. But what they do not know is the many hours of preparation that go before the actual defense by you. And then if you have 2 pages of questions and you're only allowed to ask 2 questions, yeah, then you're thinking, Well, to be honest, it all takes time. And this is time that you could otherwise spend with your families. So that is why I would like to sincerely thank you on behalf of myself, on behalf of Wannes, but also on behalf of the other promoters for all the time you have invested in Wannes and in our group. Christel, Nico, ik blijf tellen, ik denk dat dit nummer 5 is die we afleveren als een sandwich PhD met de Universiteit Gent. Nummer 6, 7 en 8 zijn onderweg. Die gaan binnenkort ook hun PhD's verdedigen. Dus ik denk dat dit een succesverhaal blijft. Ik heb het al een paar keer gedaan en ik wil dit blijven publiekelijk doen. En dat is mijn uitdrukkelijke dank betuigen aan jullie steun over de jaren heen. Steun in onderzoek dat we doen. Steun wanneer ik vraag om promotor te zijn en jullie eigenlijk zonder een seconde na te denken telkens ja zeggen. Blijkbaar hebben jullie ergens een zwakke plek voor een gekke tandarts met bizarre vragen en die ook nooit jullie wetenschappelijke uitleg snapt, maar die doet alsof. Of je voorziet dat je tandproblemen gaat hebben en dat je mij binnenkort nodig gaat hebben. Dat kan ook zijn. Maar we delen alleszins onze liefde voor die gekke orale bacteriën. En voor dat, beste vrienden, zoals ik jullie zie, mijn dank uit het diepste van mijn hart. Dit gezegd zijnde, weet ik dat er nog heel veel mensen bedankt moeten worden. En vele mensen ga ik vergeten als ik daaraan begin. Dus ik ga dat aan dokter Van Holm overlaten, zodat niemand mij kan verwijten, tenminste, dat ik iemand zou vergeten zijn. En sorry, Wannes, dat is natuurlijk het voordeel van promotor te zijn en nog heel even je baas te zijn. Wannes, onthoud dat het nastreven van kennis een nobele en levenslange onderneming is. Terwijl je verder gaat in je academische en professionele carrière. Je prestatie van vandaag is een bewijs van je toewijding en je capaciteiten. Blijf daarin geloven, ondanks wat ze vroeger gezegd hebben tegen jou, maar ook tegen mij indertijd. Je hebt bewezen dat je het in je hebt om het verschil te maken weer al in de wereld, globally. We kijken er naar uit om getuige te zijn van de impact die je ongetwijfeld gaat blijven maken. Gefeliciteerd, dokter Wannes Van Holm, met deze uitzonderlijke prestatie. Mogen je komende reis gevuld zijn met een aanhoudend succes, vervulling en een blijvende erfenis van bijdrage aan de orale microbiologie. We zijn enorm trots op je en we zijn benieuwd waar je grenzeloze nieuwsgierigheid en passie voor leren je volgende keer naartoe zal brengen. Ik kan alleen maar hopen, wij kunnen alleen maar hopen, dat wij daarmee getuigen mogen van zijn. Bedankt, Wannes, en nogmaals gefeliciteerd.
Ja, top. Okay. I'll try to do it both in English and in Dutch a bit. Dank u, Wim, voor de verantwoordelijkheid aan mij door te geven om iedereen te bedanken. Ik hoop dat ik de meeste van jullie heb kunnen includeren, maar ik heb niet foto's van ons allemaal samen. Dus ook al sta je niet op, even if you're not in the presentation, I still really enjoy our time together. But let's first start with the actual thank you. So, this being the marriage, the bruidegom, what's it in English again? the groom, this bruidegom, also has to say a few words. Many of you know me as a man of too many words, so I'll try to keep it short this time and thus break my tradition. Now, I first want to thank everyone for being here and listening in online, but especially the jury that sits in front of me. A very important part of a doctoral defense is the evaluation throughout the years, but especially this milestone in my career. Thank you so much for being here. I really appreciate it. Also, thank you, Professor Van Meerwijk, for chairing this session, and Bjorn for helping me prepare everything around this, as well as Lieve. For all of those I just mentioned, I also have a small non-verbal thank you, one that disappears a lot quicker and one that stays good for a bit longer. Secondly, I would also like to thank all of my colleagues, everyone in the lab, Centraf, and our collaborators. I tried to include you. I missed a lot of people, unfortunately. But thank you for creating such a nice working environment. This helped me in doing what I love and what we all love doing, and also having a little bit of fun outside of work hours as well. And finally, I also want to thank my family and friends for supporting me throughout my life and my journey as a PhD. I'm truly blessed by everyone being here and being in my life. Thank you, thank you, thank you so much. I think I've already said a few thank yous, but thank you, thank you, thank you. Yeah, words cannot describe my gratitude. Thank you all, allemaal.